here's my drawing meiosis video. I just have a plain piece of paper. I'm going to fold it in thirds like you would fold up a letter. The goal is to make 6 eventually. So we're going to fold it in thirds. And then we're going to fold that in half. And then I'm going to fold that in half. So that I have 2 columns of 6, or however you'd like to do it. Open this up. Ooh. Flatten this baby down. There we go. So on this side, I'm going to do interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis. On this side, I'll do prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis. I just wrote down the first letter. If you'd like to write out the whole thing, knock yourself out. So, in interphase, we have a cell. We have a nucleus. So I have red and blue pens for my DNA. An interphase doesn't really look like much, so I'm just going to draw a blue squiggle and a red squiggle. This cell has 4 chromosomes, so I'm going to have 2 reds and 2 blues eventually. Interphase is the same as it was in mitosis. It doesn't look like much, but there's molecules being built in there. Prophase 1. Here's our cell again. It doesn't matter if you don't draw it exactly the same each time, because heaven knows I won't. Now, in prophase 1, the pairs match up. So I need to match up a red and a blue together. So I'll have a blue and a red. And let's do it the other way around down here. A red and a blue. So there are 4 chromosomes total, 2 blues, 2 reds. And also we can add in the beginnings of our spindle here. So this would be my centrioles. The nucleus is still kind of here, so you can like draw a dotted line around if you want, because it hasn't quite gone away all the way. I'm not including prometaphase just to make this go a little quicker, but it would be in there too. That's where the spindle would attach. Metaphase. Here's my cell. So now I take those chromosomes and they come to the middle with their partner. It's not a very good middle, but I got pretty close. Here's one side, other side. Now my spindle, 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 spindle. In here, in prophase 1 is where crossing over would happen. Here is where independent assortment would happen. So you can see I have red, blue, and blue-red on each side. So it could be blue-blue on the left and red-red on the right, or it could be mixed up. It just depends on which way they feel like going. They decide. Anaphase. Sorry, I'm dropping my pens. They're going to get pulled apart. And I'm going to draw one of these pairs being crossed over a little bit. So let's draw out this one. There's a little bit of blue on that red, and a little bit of red on this blue. Let's draw my spindle again. Telophase, I'm just going to draw kind of like an oval. And draw them a little further apart because the cell's starting to move. Yeah. Well, you could probably just draw telophase and cytokinesis together if you want. And cytokinesis, we're going to get 2 cells. One cell has a plain blue and then a red with a little bit of blue on it. The other side has a plain red and a blue with a little bit of red on it. So far, we've made a copy of the cell we started with. Now, when I come over to here, there is no second interphase. So if you want to, you can just kind of go like this. Does not exist. There isn't one. It doesn't go through interphase again. Prophase 2, now we're basically going to redraw what we just drew for cytokinesis because prophase 2 isn't that exciting. Whoa, that was awful. My cells are getting worse. Okay. Blue, blue, red, red. You could draw as much crossing over as you want, really, but I'm just going to switch 2 pieces to keep it from getting too confusing. Here's my new centrioles and my 2 new cells. Trying to form. So it's almost the same as this one, except there's no crossing over happening. Metaphase 2, again, we need 2 cells. These babies are going to line up down the middle, which I kind of already have them in the middle. Get my crossing over from before. Spindle attaches. Oh my goodness, my drawing's getting worse as we go on, guys. It's not a good sign. So we have cells in the middle in both cases, and also in mitosis, they lined up in the middle. It just, they did something kind of fancy here, but metaphase always means in the middle. Anaphase. They're going to pull apart. So I'm going to draw them like this because it shows them kind of getting pulled in the middle, like they're bent over. Which side had the red? Say this side had the red. This side had the blue. Okay. Telophase, same idea as what we did over here. We're just going to draw it a little bit further apart. So I have oval, oval, blue, blue. Making sure I keep my crossing over consistent. And now, we made it to the second cytokinesis. Now we're gonna make 1, 2, 3, 4 new cells. 1 cell, 2 cell, 3 cell, 4 cell. Okay, so this first cell gets a piece of blue and a piece of red that came from right here. Second cell gets a blue and a red that has a little bit of blue stuck on it that came from right here. Red, red, blue, blue. So you can see that all of those cells are different combinations of each other. We end up with 4 different cells in the end, which was the point, for them to be different and have half of the chromosomes. They're half of what we had before. So overall, there's the top part of it, the bottom part of it. My iPad screen's only so big, sorry. The takeaway here is the crossing over and independent assortment that happened here end up with us having genetically different end results. And we have to go through this twice, so that way we end up with haploid cells that have half the chromosomes. If we only did it once, they wouldn't be haploid. Make sense to everybody? In case you're thinking of this, because I know some of you are good at math, you're thinking about, but this was 4, and this only had 2 down here, so wait a minute. Remember in interphase, the DNA gets copied? So we would have 2, but then it got copied into 4 and split in half. So we started with 2 and we're ending with 2 at the bottom. Don't think too much into the math. And then we take those ones and divide them in half. So we started with 2 full chromosomes and ended up with halves of chromosomes here. Make sense? I hope so. We will come back to this idea when we talk about genetics, so if you're super duper confused, please let me know. Let me know how I'm doing. See you next time, guys.